Hello friends, I welcome to you all TIBSC students in our today's lecture. As you know that from semester 3rd actually computational botany paper, that is sixth paper, is being taught to you by me. Today we are going to learn unit 3rd, analysis of data on vegetation studies. Students, actually studies on vegetation began in early 19th century with the work of Alexander von Humboldt. Actually, in 19th century, in the sense 1805, this is the actually exact year of 19th century in which the studies on vegetation began. Now, this field work conducted to study the distribution of taxa and their evolutionary relationship. Over the time, approaches have developed towards the vegetation study in different areas of specialization. The vegetation cover of the earth is extremely complex, and thus all vegetation's data gathering must be the selective, since it would be impractical to undertake total recording or description of vegetations. As we know that detailed vegetation study is based on description and investigation of the plant community, identifying the appropriate sampling technique first requires for such a type of a study. To do this, the researchers should consider actually objectives of the study, life forms, distribution pattern of individuals, of a species, distribution pattern between species, that is community mosaic pattern, efficiency of the data collection from the economic standpoint, and accuracy and precision of the data is required. Friends, why I'm giving idea of all these things before getting idea about the calculation of collected data. As we know that data is obtained from the quadrats and transect methods. I know very well, you know very well, in the last year SYBSc, there was one of the practical, that is study of vegetation by using a quadrat. Now, why these techniques are to be used? What is the goal of this one? This study. The goal of this study is that restoration and revisitation project is to create the plant cover distribution and species composition of the site prior to disturbance or of a comparable less disturbed reference site. Friends, you will have to keep in mind actually the United Nation or the ecologist or the botanist or the scientist or environmentalist used to say that there should be the 33% green cover should be on the earth. But right now, anybody knows about that one? Only 11%, sorry, only 17 to 18% is there. So there is a too much lacuna, isn't it? It's near about, if we'll say that, 15% lacuna is there in case of the green cover, and because of that one, many more problems are being faced by the human community right now. And there is a lot many things that you won't know very well. There is endangered species, the species extinction, organism extinction. So such a things are happening because of that one. But that can be saved by collecting the data by using some quadrat method or transects method. Now, why to use the quadrat methods? Isn't it? Why to use the contract method? Actually, it is impractical to take a complete census of even a relatively small site. Cover density and frequency of the plant species can be accurately estimated from as little as 1% of the community. In the sense, suppose anybody asked you go and study the vegetation of forest. We cannot study that huge area having forest. But sampling is possible, and that sampling means 1% area of that forest can be studied very well with the help of a quadrat method. That quadrat method gives us idea about kind of plants in a particular habitat, number of that actually plant species. Also we can understand with the help of a such type of a quadrat methods. Now this is, I have to say that all about the quadrats, isn't it? Transect and quadrats are two ecological tools that allow us to quantify the relative abundance of the organism in an area. To track the changes over the time, it is important to be able to quantify the changes in abundance. Also, learning these techniques will give the idea to the students the tools necessary to ask their own ecological questions and make comparison among the sites. In the sense, we know that suppose once we have visited to the forest, we have collected the data, that is not enough. Frequently that area, that habitat of the plant is to be visited and data is to be collected, so that we will come to know that the changes going to take place into that one. Abundance of the species, again rare species. So if suppose rare species we have found, definitely we will be make some efforts just to increase their number. So that is actually use of all this quadrat method or transect method of study. Now a transect is a simple line we stretch over an area we want to study. Transect method is very simple. Only we have to sketch one line, and along that line, along on that area, only we have to study. The line must have a regular measurement marked up like a tape measurement and is held straight and stationary. That is the, I have to say that the transect method is there. A quadrat is framed area that you have studied in the last year. Framed area. A frame can be made using PVC pipes, wire hangers bent into the squares, wooden dowels, or even a cardboard can be used to prepare the quadrat. That is a square, 1 square meter area. Use monofilament or strings to section off the quadrat into a set number of squares or intercepts. In the sense, 1 by 1 square meter area is there. That is, we have to divide into a small, we have to say that squares for our study. So total that 1 by 1 meters area, we have to divide into 4 quadrats, larger size, isn't it? And then we can study the vegetations. Now, after collecting data with the help of a quadrat method or transect method, we should have knowledge about some formulas, what we want to calculate. After calculation, what we have to do. So here, percent frequency, how to calculate that one. In case of percent frequency, here my friends, number of quadrats in which the species occurred, here word should be the species occurred, isn't it? Number of quadrats in which the species occurred. In the sense, suppose we have set up there 4 quadrats and species occurred into a 3 quadrats one. So we have to write number of quadrats in which the species occurred, only 3 is there, isn't it? Total number of quadrats studied, that is 4. So 3 by 4 into percent, that is multiplied by 100. Species occurred into only 3 quadrats. We have set up only 4 quadrats multiplied by 100. That is percentage frequency we can calculate. Now, another we can calculate, that is a relative frequency. Relative frequency, the formula is here, number of occurrence of species, that is total number, and number of occurrence of all the species. So suppose number of occurrence of species is a 10. Occurrence of all the species, that may be the more than 10, that is 100 multiplied by 100. So 100 in only 10 is the relative frequencies there of that specific single species. I am giving you idea of all this. Now relative density, total number of individuals of species in all quadrats. Total number of quadrats studied. So total number of individual species, suppose Parthenium histoporus, गाजर गवत, काँग्रेस आहे, ते आपल्याला किती सापडलं टोटल? तर समजा एक 100. टोटल नंबर ऑफ कॉड्रॅट स्टडीड, मगाशी आपण 4 घेतले तर 4च घेणार, 4 आर दॅर, isn't it? Total number of so that could be the relative density will be there, isn't it? So this relative density, relative frequency we have studied, relative density we have studied, isn't it? So this is not actually relative density, this is a density only. Relative density formula is given here. The relative density is to be calculated by number of individual species divided by number of individuals of all the species into 100. That is this formula. Here the formula before relative density we have studied, that is okay. Density you have to keep in mind, that is total number of individuals of the species in all chordates divided by total number of chordates studied. Parthenium histoporus 100 आपल्याला सापडलं. किती क्वाड्रॅट आपण स्टडी केले? 4. येणारं उत्तर असेल दॅट इज अ 25. 25 इज द डेन्सिटी ऑफ द पार्थेनियम हिस्टोफोरस, isn't it? Now relative we have to multiply it by 100. Number of individuals of the species 100 आहे. Number of individual species of number of individuals of all species सगळे, मग त्याच्यामध्ये सगळे जे पार्थेनियम हिस्टोफोरस आहे, मे बी स्पोरोबोलस grass is there, maybe we have to say that the Boravia diffusa plant is there, maybe we have to Argemone mexicana is there, isn't it? Maybe Dudhni, isn't it? So that type of plants are there. त्याचा नंबर किती आहे? That may be the, suppose 199 is there, multiplied by 100. That is the relative density of the Parthenium hysterophorus. Density only number of individuals divided by chordates in which that is occurred, that is equal to 25% is there. So this formula you have to always keep in mind. Solve some examples, then only it is possible to understand that. Now abundance, isn't it? How to calculate the abundance? Very simple methodology. Total number of individual species in all the chordates. आपण परत पार्थेनियम हिस्टोफोरस 100 घेतोय, isn't it? And total number of chordates in which the species occurred 4, isn't it? That is a 4. So that density and abundance is 25%. Dominance, how to calculate the total area of a given species per unit area within a community? That is nothing but the dominance. Relative dominance, total basal area of the species divided by total base area of all species. म्हणजे काँग्रेसचा बेसल एरिया किती आहे, ऑल स्पेशीजचा किती आहे, मल्टिप्लाइड बाय 100, that is equal to relative dominance. That is equal to relative dominance, isn't it? So these formulas you will have to keep in mind, you have to study very well. With this, thank you very much. Again I will give advice to you, my friends. You will have to first of all read the unit 10 very well. Unit 10, that is analysis of data on vegetation studies, and then you will come to know that how the things are going to happen, and then you will have to get study about the simple ratio or ratio vegetation index, RVI. What is RVI? Simple ratio index, SR, isn't it? Difference vegetation index, DVI, normalized difference vegetation index, NDVI, or greenness index, isn't it? So all this information you will have to again study, means computation of a crop or vegetation biomass using a satellite data. As just now we have seen how that is calculated data with the help of we have to say that the quadrats and transit method. Now new method, which is a fast accurate method, is there, that is of a satellite data, computation of a crop vegetation biomass using a satellite data. So with the help of that satellite data, immediately we can measure a huge area get accurate results, and that also is there in that unit 10 that you have to prepare on your own. Okay, thank you very much.